Let's go over the basics of amino acid metabolism, how we can safely remove those amino groups and use the parts from amino acids to do cool stuff without having our bloodstream build up with ammonia because we don't want Windex veins. So instead, we'll use the urea cycle in the liver to safely dispose of those nitrogen groups as urea. We'll often transport those amino groups to the liver in the form of glutamine or alanine. We'll go into each of these points in more detail. However, here are some key things to keep in mind as we go through. First is that transamination is the process of transferring an amino group from one molecule to another. If we transfer an amino group from an amino acid, what we're left with is an alpha-keto acid. So our amino acids are alpha amino acids because the amino group is on the alpha carbon, the carbon that's alpha to the carboxylic acid group. In the case of removing that amino group, if you think about putting a ketone in the place of the amino group, well, now you're going to have a ketone in the alpha position. So this is an alpha-keto acid. So if we remove an amino group from an amino acid, we get an alpha-keto acid. And if we add an amino group where the ketone was on an alpha-keto acid, we get an amino acid. And these reactions are going to be carried out by enzymes called transaminases. Often with amino acids, we're going to be transferring an amino group from an amino acid onto α-ketoglutarate to give us glutamate. This is going to leave us with the skeleton of the amino acid from which we transferred the amino group. So for example, say we were to transfer an amino group from alanine onto α-ketoglutarate. This would leave us with pyruvate and glutamate. Often we're going to be making glutamates because then we can kind of have transferases that are going to specialize in using glutamate to pass the amino groups from glutamate onto other molecules. So for example, if you took a pyruvate, you could have an alanine transaminase pass an amino group from that glutamate onto pyruvate to give you alanine. And this way you'd be able to make alanine from any amino acid if you transferred the amino group from the first amino acid onto α-ketoglutarate. Now you have this enzyme alanine transaminase that can take that amino group and pass it on to pyruvate to give you alanine. Similarly, aspartate transaminase can pass an amino group from glutamate onto oxaloacetate to give you aspartate. So we can interconvert amino acids. We can take the amino group from one amino acid to make another amino acid. We can also pass the amino group from one amino acid to an alpha-keto acid in order to just get the carbon skeleton from the first amino acid that we can do things with. Transaminases are often going to use a cofactor called pyridoxal phosphate to carry this out. So we've seen a lot of cofactors. So we've seen like biotin was really great for carboxylation. We saw NAD was really great for oxidation. And now we're seeing PLP, pyridoxal phosphate, is really good for transferring amino groups, as well as for some other things. What's basically going to happen is that you can have this thing called a shift base, where you have this imine group where you have a nitrogen double bond to a carbon, and that's kind of like swappable for a carbonyl. And so what happens is that the pyridoxal phosphate can actually make a bond, one of those shift-base bonds, to a lysine in the transaminase active site. This is going to hold the PLP in place. Then what's going to happen is that an amino acid is going to come in, and it's going to kick back off the lysine and hook itself onto this pyridoxal phosphate. So now this pyridoxal phosphate isn't like directly attached to the enzyme anymore, but it is attached to the amino acid, and the enzyme is still holding it onto it. So it's basically still held in the enzyme. Now what you need to do is you need to cut off the amino group. And so then water can come in and cut that off. Now you transfer this amino group onto the pyridoxal phosphate, which was being held by the enzyme. And you've released an alpha-keto acid. So we've taken an amino acid, we put the nitrogen group onto this pyridoxal phosphate. This gives us a molecule called pyridoxamine phosphate, PMP, and it leaves us with the alpha-keto acid. If we want to now move this amino acid to something else, well, now we just have to bind to that something else, bind to an alpha-keto acid, so in this case, alpha-ketoglutarate, transfer that amine group to the alpha-ketoglutarate, and then rebind to the enzyme's lysine. So because you have this kind of like swappage point on the PLP, you're able to variably make bonds to either nitrogen or to an oxygen. You can swap one from the other, forming a bond to the nitrogen in the active side of the enzyme, then forming a bond to the nitrogen in the amino acid, and then forming a bond to oxygen from water. And this is then going to restore your PLP. You could do this again and again. We'll talk about how there's a lot of cool things you can do with amino acids, including making glucose and making lipids, depending on the properties of the carbon skeleton that you're left with. However, we need to dispose of those amino groups safely if we want to avoid the buildup of ammonia. If we were to just cut off the amino group as is, this NH3 or NH4 plus, if it were to protonate, that would be ammonia. Ammonia is toxic. We don't want ammonia building up, so instead we're going to transport those amino groups through the bloodstream to the liver, where the liver is going to carry out something called the urea cycle. I don't need you to worry about the details, but I will walk you through it one time. The key thing to focus on is the fact that you're going to be taking two amino acids that travel to the liver on the form typically of glutamine or alanine, as we'll discuss. In the liver, these amino groups are then going to get incorporated into this molecule called urea and get excreted. For each turn of the urea cycle, you're taking two amino groups and you are removing them as urea. This is also going to leave you with carbon skeletons of the amino acids, and then we get into talking about what can we actually do with those carbon skeletons. For each turn of the cycle, we're going to need to bring in two amino acids because we're going to need two amino groups to make urea. These two amino acids can be coming from the bloodstream, and remember, those could be coming from any amino acid. Typically, then the amino groups will be passed on to make glutamine or to make alanine that transfer to the blood to your liver. In the liver, one of these amino groups is going to get passed on to α-ketoglutarate to give you glutamate. And then this can happen in the mitochondria or this can happen in the cytoplasm, but one way or another, that glutamate is going to get into the mitochondria. And then the amino group is going to get released as ammonia. We'll get back to this in a minute. The second amino acid is also going to transfer an amino group onto α-ketoglutarate to give you glutamate. But now this glutamate is going to get passed onto oxaloacetate to give you aspartate. So you kind of have these two pass-offs. These transaminations can happen in the cytoplasm or in the mitochondria. And so you can have the amino acids travel through. And therefore, these transaminations can happen either place, but one way or another, you've got to end up with a glutamate in your mitochondria and an aspartate in your cytoplasm, because part of the cycle is going to take place in the cytoplasm, the part that takes the amino group from an aspartate, and then the other part, the part that takes the amino group from a glutamate, is going to happen in your mitochondria, or at least the part where it takes the amino group from a glutamate with an oxidative deamination that we'll talk about. Whereas here, we're doing a transamination. Here, we're going to do something called an oxidative deamination. And so we'll talk about that. But remember that you have two amino acid groups coming in, two amino groups being passed. Those two amino groups are ultimately going to be released as urea. And now we just have to figure out the logistics to make that happen. Note too that in the next few minutes of the video, I accidentally was using a figure where the NAD plus and the NADH were switched. Remember that malate reacts with NAD plus to give you oxaloacetate and NADH. Oxaloacetate is the more oxidized form. This is an oxidation reaction going from malate to oxaloacetate. It needs NAD plus. The reverse, going from oxaloacetate to malate, is a reduction reaction that uses NADH. So I wasn't able to fix in post-production the swappage on those couple slides or couple minutes or whatever because I was moving the screen around. So thanks for understanding. It should be like this. So we have aspartate, and so that was made with aspartate transaminase, and then we have our glutamate, which will be made by various transaminases. So we have two nitrogen groups, one on each of these, and we can have gotten those amino groups from any amino acid. So what we're going to do is that this glutamate is going to get oxidatively deaminated. So you're going to do an oxidation reaction to cut off that nitrogen-containing group as ammonia. Overall, we kind of call this process transdeamination, We're transaminating from an amino acid onto alpha-ketoglutarate to give us glutamate, and then oxidatively deaminating the glutamate to give us alpha-ketoglutarate and our ammonia. And yes, we don't want free-floating ammonia hanging around, but this is happening in our liver, in the mitochondria, in this controlled manner where it's going to quickly react with bicarbonate in this reaction that uses ATP and generates this highly energetic molecule called carbamoyl phosphate. That's happening with the help of carbamoyl phosphate synthetase 1, which is going to be a regulatory point, kind of like the main regulatory point, other than just controlling concentrations. Its production is regulated by arginine. So this whole process is kind of indirectly regulated by arginine. You have this molecule called carbamoyl phosphate, which if you look at it, what we have here is kind of this activated amide. So remember a nitrogen group next to a carbonyl, that's an amide. And now it's attached to a phosphate group, so that's going to be a kind of place at which we can link this up somewhere else. Where we're going to link this up somewhere else is onto the end of this molecule called ornithine. If you look at ornithine, well, it looks like arginine, but it's missing kind of the end group of arginine. If you look at the end group of arginine, well, that looks a lot like urea. If you just think about kind of like adding a water and chopping this end off of arginine, you get urea, and you're left with ornithine. This is what we're going to do. But in order to actually make the arginine, what we're going to need to do is we're going to need to add two amino groups. And we're going to get one of those amino groups from our glutamate that we have coming in over here, and then one of the amino groups from our aspartate. So what's going to happen is that the ornithine is going to join with the amide group from our carbamoyl phosphate. And then this is going to, with the help of ornithine transcarbamylase, and this will give us citrulline. Now the citrulline can transport out of the mitochondria, and this next part is going to happen in the cytoplasm. So we're going to see this kind of antiport, this co-transport, where citrulline is going to get passed out, ornithine is going to get passed in, this ornithine carrier translocase. The citrulline now in the cytoplasm can join up with the amino group from aspartate. And we're going to do it in this really awkward manner using this molecule called arginosuccinate synthetase. As the name suggests, we're going to be synthesizing a molecule called arginosuccinate, and this arginosuccinate is this really, really awkward looking thing. Basically, what's happening is that instead of transferring the amino group like we've transferred amino groups from place to place, we're actually going to kind of stick this whole aspartate onto citrulline, onto this end of citrulline, in the place of this, where this carbonyl is. We're going to stick on this whole aspartate. And this is going to give us this arginosuccinate. It's going to be energetically costly. It's going to cost us an ATP, but it's almost like it costs us 2 ATP because we're hydrolyzing it to AMP rather than ADP. So it would take us 2 ATPs in order to regenerate this. But we end up with this molecule, arginosuccinate. This molecule does not like being this awkward, and so arginosuccinase is going to help us kind of like break off the part of the aspartate except for the amino group. So that'll give us fumarate. And then we can take the rest of it. So now we have the amino group added on. We have our arginine. And now we can break off the end of arginine like we were talking about before. We can hydrolyze it with arginase. Use water to kind of break that off, give us urea. That urea can now go to your kidneys and get excreted in your urine. What you've done is you've taken 2 amino groups and you safely removed them as urea. Voila! You're also left back with ornithine that can go back into the mitochondria in exchange for new citrulline, and then you could do this process over and over. Each time you're going to be bringing in 2 amino groups, which are coming from a variety of different sources, but then kind of come from glutamate or aspartate. But the amino group themselves could have been transferred there from pretty much any amino acid. So let's think about how we can keep this cycle going. Remember that we had those 2 amino acids bringing in amino groups, and then those amino groups are going to get passed on to alpha-ketoglutarate to give you glutamate. Ammonia was going to be taken from one of those glutamates, leaving you back with alpha-ketoglutarate. This alpha-ketoglutarate is now ready to accept another amino group, and so we're good on that front. What else are we going to need, however? In order to pass this amino group on, another amino group on down here, we're going to need another α-ketoglutarate in a similar situation. However, when we take the amino group from the glutamate and then take it onto aspartate, we then take the whole aspartate and add that onto citrulline. We're not just removing the amino group. So we're actually taking all these carbons into the cycle, and then those carbons get removed as fumarate. So we're going to need another oxaloacetate if we want to keep this going. We need an oxaloacetate where we can pass the amino group onto in order to give aspartate. We regenerated our α-ketoglutarate so we can make glutamate up here, and we're going to regenerate this α-ketoglutarate here when we pass the amino group onto oxaloacetate to make aspartate. But then we're going to take that whole aspartate away, and so we won't have this oxaloacetate regenerated. So we need to regenerate that oxaloacetate. In the citric acid cycle, we know that we can go from fumarate to malate using fumarase, and then malate to oxaloacetate using malate dehydrogenase. And we actually have those enzymes in the cytoplasm, but in the citric acid cycle, they're happening in the mitochondria. So if you want to use this fumarate to make oxaloacetate in the cytoplasm, you can, but that's not going to be a simple link to your tricarboxylic acid cycle. So although sometimes you might see this thing called like Krebs bicycle, where you have oxaloacetate siphoned off from the citric acid cycle connected to the urea cycle and just have this like continuous loop, although that like energetically works in terms of just kind of keeping a balance and a figure on a piece of paper, it's not practically useful because the fumarate is in the wrong place. We don't have transporters for fumarate to get into the mitochondria. So the fumarate is not just going to be going into the mitochondria, going through your citric acid cycle, and then you take the oxaloacetate out. You could make the oxaloacetate out in the cytoplasm and use it there. But there's probably better things you want to do with that oxaloacetate that you make out here. Because you have this oxaloacetate out here and the liver has other things it can do with the oxaloacetate. The liver can take this a step further. The liver can do glucogenesis. It can use PEP carboxykinase to turn that oxaloacetate into phosphoenolpyruvate and then take that through glucogenesis to make glucose. Why does this make sense? Well, if you think about why you would be breaking down proteins and running the urea cycle, there's a couple of different scenarios. One is that you have just eaten a big meal, you have a lot of protein, and therefore you're going to want to store the carbons from that protein as glycogen. And so you can take that glucose-6-phosphate, you can do glycogenesis, link that up into glycogen and store it. Another situation is that you're breaking down proteins because you need energy, you're starving, and in this case, the liver, it needs to supply glucose to the body. So it can take that glucose it makes and it can ship it out as glucose. And it can ship it out to the tissues who need it. So great, the liver is able to do its job, remove the amino groups safely, and give fuel to the body or store fuel. Now you might be wondering though, weren't we trying to figure out how to keep this cycle going? Yes, but there are also the carbon skeletons left over from those amino acids that you brought in. So our main amino acids that carry nitrogen groups through the bloodstream are going to be glutamine and alanine. So your muscles have this cool glucose-alanine cycle, and your other tissues are going to mainly use glutamine. So imagine you have an alanine, and you remove the amino group. That's going to give you pyruvate. We know that pyruvate carboxylase can take that pyruvate and make oxaloacetate. We need oxaloacetate for the cycle. Voila. Because we can do the transamination in either the mitochondria or the cytoplasm, because we can take aspartate in or out of the mitochondria. We just can't take oxaloacetate out in or out of the mitochondria. So we can make oxaloacetate in the mitochondria, transaminate it in the mitochondria, take that out of the aspartate out of the mitochondria, and then do the cycle out here. Or we can make the oxaloacetate in the cytoplasm and do the cycle over here. So we have options for how we get that oxaloacetate. And you're typically in your liver, it would be getting it from the skeletons of the amino acids that it brought in, rather than just taking this fumarate and getting the oxaloacetate from it. Because that oxaloacetate it gets from it is often going to be better used by the liver in order to do things like glucogenesis and to deliver glucose to the body or to store glucose as glycogen. because it's going to have to do something with those amino acid skeletons anyway. And if it's already, you have this fumarate out here where it's needed, where you have these enzymes that can help make it glucose, that's going to be a better step, less kind of gymnastics trying to get things in and out and trying to coordinate all the stuff. So not that simple bicycle, but one way or another, you are going to be able to regenerate that oxaloacetate to keep that cycle going. You don't need to worry about regenerating the ornithine or the citrulline because those are getting regenerated in the cycle. The only things that you need to kind of worry about getting back is this oxaloacetate. And whatever you do, you're going to be left, though, with the carbon skeletons from those amino acids that you deaminated. So you're going to be left with an alpha-keto acid, where there's a ketone in the place of the amino group. Depending on the amino acids we started with, we'll be left with different alpha-keto acids. The alpha-keto acid can then go through various processes depending on the amino acid. It might be something that can enter our citric acid cycle. And if it can directly enter the citric acid cycle, great. If not, we can go through a couple steps to get it there. We can transform it into other molecules. Speaking of getting into our citric acid cycle, if we have a molecule that is able to enter the citric acid cycle and it's more than 2 carbons long, that's going to allow us to have an anaplerotic reaction, basically. It's going to allow us to replenish our cycle, which is good because, well, if we're taking oxaloacetate out of the cycle to do this process potentially, then we're going to want to add stuff into the cycle. And so we can often use the skeletons from those amino acids in order to feed into the cycle. And because we could take malate to make oxaloacetate to make glucose out of the cycle, if we then replenish the cycle with an amino acid, we say that amino acids that are the skeletons from amino acids that can enter the tricarboxylic acid cycle, the TCA, and it's more than 2 carbons long, are going to be what we call glucogenic, because we can sustainably make glucose from them. However, products that we get from amino acid skeletons that are only like 2 carbons, so they're only like acetyl-CoA, that's not going to be glucogenic because each turn of the cycle we're removing 2 CO2, we're removing 2 carbons, so we need to put in more than 2 carbons in order to keep the cycle going sustainably if we take stuff out of the cycle. So that's our whole differentiation between our glucogenic and our ketogenic. With our glucogenic, it's going to be things that you're adding in that will allow you to sustain the cycle if you take carbons out. And so since you take 2 carbons out each turn of the cycle, you're going to need to put in more than 2 carbons. And if you only are making acetyl-CoA or acetoacetate or one of those ketone bodies, you can use those to make fats, and you can use them to make ketone bodies, so we call those ketogenic, but you're not going to be able to use those sustainably to make glucose, because if you put the carbons from acetyl-CoA into the pathway, you're only putting 2 carbons in, and you need to put more than 2 carbons in order to keep this cycle going as a cycle if you are taking things out of the cycle. So you got to give 2 pennies to each turn to take 2 pennies each turn. If you take out a nickel, you got to put in a nickel. And the urea cycle was a way that we can safely dispose of the amino groups from those amino acids so that we could use the skeletons from those amino acids to do cool things, like make that glucose, like make that lipid. For all of those things we want to use the carbon skeletons of amino acids for, we need to first have a way in which you can get rid of the amino groups. And so we do this by passing the amino groups off of those amino acids onto an alpha-keto acid, typically onto α-ketoglutarate to give us glutamate. And then we can take that glutamate and we can take the amino group off of it and incorporate it into urea, or incorporate it into aspartate, and then we can incorporate it into urea. And this way, we're able to remove those amino groups. Note too that when we're passing these amino groups through the bloodstream, it's often going to be in the form of alanine or in the form of glutamine. And then once those get to the liver, then they can kind of pass that amino group off onto α-ketoglutarate to give you glutamate. And then that glutamate is kind of like the main one that can take care of swappages from there. A lot of your tissues are going to use glutamine to transport that amino group, but your muscles and your liver have this sweet deal, at least sweet for the muscle sense, the glucose-alanine cycle, where the muscles send the amino groups off in alanine, and then the liver gives them back glucose. So like we saw in the Cori cycle, the muscles are kind of making the liver do a lot of hard work often. And so in the case of the Cori cycle, the muscles are breaking down glucose to give pyruvate through glycolysis. And that was leaving them with NADH, but they didn't have enough oxygen and all that stuff to go through the citric acid cycle and the electron transport chain. And so then the NADH would build up. And so instead of letting the NADH build up, the muscles would go through lactate fermentation. They could use that NADH to reduce pyruvate to lactate, but then they're left with the NAD+, which they can use for glycolysis, but they're also left with the lactate. So they ship the lactate out to the liver, and the liver converts it back to pyruvate, and then converts that to glucose and sends that to the muscles. Awesome. In this case, we are not dealing with any oxidoreduction, but we are dealing with the muscles making the liver do stuff. You can consider that the muscles are breaking down some protein, and when the muscles are breaking down that protein for those carbon skeletons, they're going to need to do something with that amino group. And they want to get that amino group to the liver. And not only do they want to get the amino group to the liver, but they also want to get stuff back in return. Because the way they're shipping that amino group to the liver is in the form of alanine. If they're doing this, they're taking the pyruvate that they get from glycolysis, and they're transferring glutamate, an amino group from glutamate, which remember they could get from basically any amino acid onto glutamate, and then alanine transaminase can put that amino group from glutamate onto pyruvate to give you alanine that they can ship out. Normally this pyruvate, if you wanted energy, you take this pyruvate through the citric acid cycle and you would get energy that way. However, if you then ship carbons from this pyruvate out in the form of alanine, you're losing that opportunity. And so what you want to do is you want the liver to send you stuff back. And more, you want the liver not just to send you alanine back, but to send you a whole glucose back. So it's like with the Cori cycle where we're sending out lactate and getting back glucose. So we send out 3 carbons, we get 6 carbons back. Here we're sending out alanine and getting glucose back. So we're sending out 3 carbons and getting 6 carbons back. The difference in this case, or one of the differences, is that we're also sending the amino group and we're not getting that amino group back. The liver's got to take care of the amino group, but the liver's got the urea cycle, so the liver can do that. So the muscles send out this alanine, and then the alanine gets taken into the liver. The liver can then take the amino group off of alanine using alanine transaminase. So just like we saw our muscles use alanine transaminase to aminate pyruvate to give you alanine, in your liver, you're going to aminate α-ketoglutarate from alanine to give you glutamate. So you take that amino group back off of alanine, back onto α-ketoglutarate. That'll give you your glutamate, which you need for the urea cycle, and it'll leave you with pyruvate, which the livers can then go through glucogenesis to make glucose to send back to your muscles. So this is called the glucose-alanine cycle. The muscles use alanine transaminase to transaminate pyruvate to give alanine. The amino group that was on that glutamate could be coming from any amino acid that the muscles wanted to break down for energy, because we can transaminate α-ketoglutarate from any amino acid basically to give us glutamate. And then we take the amino group from glutamate, put it on pyruvate with alanine transaminase to give us alanine, ship that alanine out into the bloodstream where it travels to the liver. The liver takes the amino group off of the alanine, puts it onto α-ketoglutarate to give you glutamate with the liver alanine transaminase. That leaves you with pyruvate that the liver goes through glucogenesis to make glucose, which sends back to the muscles. It also leaves you with glutamate, which the amino group from that glutamate can then go and enter the urea cycle. And that will give you back your α-ketoglutarate that can do this again and again and again. And so the muscle's getting rid of a nitrogen. It's getting rid of 3 carbons, but it gets back 6 carbons. And so it gets even more than it sent out. And so the muscle, yet again, is making the liver do its hard work. And this is great because the muscles, if they're breaking down proteins, they probably need some energy. Quick medical note. So you see that aspartate transaminase is playing a key role in your urea cycle, which is taking place in your liver. You're also going to have a lot of alanine transamination happening in your liver, taking in those amino groups from amino acids that transferred through your bloodstream. Therefore, the enzymes alanine transaminase ALT and aspartate transaminase AST are going to be high in your liver. Typically, however, those enzymes would be in your liver and not in your bloodstream. However, if there's problems with your liver, then you can get these enzymes in the bloodstream, and that is something that doctors can measure and determine whether your liver is having problems. So if you hear of like liver enzymes, those are typically referring to alanine transaminase and aspartate transaminase, your ALT and AST levels. So I was curious about the glutamine versus alanine, and it turns out that the alanine is the preferred source in your liver, but the glutamine is the preferred source in your kidneys. This allows for things like the differential regulation of the glucogenesis, of glycolysis, all of this different stuff in your livers versus your kidneys. Just like we could break down amino acids to get carbon skeletons that could enter the TCA, we can also take carbon skeletons out of the TCA in order to make amino acids. There are a lot of amino acids that we can just make directly from the TCA or just in a couple of steps. So, for example, we've already talked about how we can convert oxaloacetate and aspartate by simple transamination, similarly with α-ketoglutarate and glutamate. We can also easily get alanine from pyruvate and various kind of other easy transitions. These are going to be examples of non-essential amino acids. So these we can make easily from scratch. There are also, however, going to be amino acids that are essential. Now, when we say that alanine and asparagine and aspartate and glutamate and serine are non-essential, that doesn't mean our bodies don't need them. Our bodies definitely need them, which is probably part of why we can make them from scratch. It's so important that we make them ourselves, and we can make them ourselves. But there are other amino acids that are really important too, and we aren't able to make them ourselves from scratch. And so we call these essential amino acids. An example of an essential amino acid is going to be phenylalanine. We need to get phenylalanine from our diet. And if we don't get phenylalanine from our diet, then we can't make it ourselves, and so we're a little screwed. If we look, some of things are considered conditionally essential. These, under certain conditions, the body might not be able to keep up, and these are therefore considered conditionally essential. You do have ways to make it, but maybe they're not that efficient. Maybe you don't have enough of the precursor, things like this. There are conditions, like not conditions as in medical conditions, like you have a disorder or disease, but just times in your life or things like this where you might need to get those amino acids from your diet rather than just making them yourself. And so one of the examples of this is going to be tyrosine. Tyrosine is made from phenylalanine, and phenylalanine is essential, so tyrosine is considered conditionally essential. So you get various cases like this, where the amino acids are being made from other things that maybe are limited, or those things are essential, and therefore they're considered conditionally essential. Whereas non-essential, we can really just make them really easily, pretty much. And we'll often see that a lot of these non-essential ones are going to be ones that we really, really rely on in our metabolism, especially in the transfer of amino groups and things like this. So like our alanine with our glucose-alanine cycle, our asparagine and our aspartate, and our glutamate, which are really important for the urea cycle, our serine, which is important for all sorts of things. So amino acids have a lot of uses, and we can make a lot of them. We can't make a few of them. So we need histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine from our diet. And then we can make the rest, although some of them we might not always be able to keep up. But all of this goes back to the idea of kind of thinking about small little pieces as being really useful because you can piece them together in lots of different ways. We've got that Lego kit. We break down those big molecules into these little molecules that we can interconvert between one another. The amino acids are a little weird in that they do have those nitrogen groups. And so we need to be careful with those nitrogen groups so that we don't have ammonia in the blood. But they are good if you want to put a nitrogen group into something. For example, glycine can be used to help make purines, so the rings of your DNA and your RNA. Glutathione, our antioxidant. porphyrins, so like the heme in your hemoglobin that allows you to transfer oxygen through your blood. All of those are relying on using the skeleton of glycine, so not just for proteins. We can also make hormones from some of these amino acids, so like adrenaline and noradrenaline. Remember, those are epinephrine and norepinephrine. Those are produced from tyrosine, which is produced from phenylalanine. We also get thyroid hormones from tyrosine, as well as melatonin, serotonin, all these really great things. For the key things, transamination is the process of transferring an amino group from one molecule to another. If we transfer an amino group from an amino acid, what we're left with is an alpha-keto acid. That will let us interconvert amino acids. It'll allow us to, say, make glutamine or alanine from the amino groups of those amino acids. We can then take that glutamine, take that alanine through the bloodstream to the liver, and in the liver, we can safely dispose of those amino groups in the form of urea. Or at least we can make the urea that then the kidneys can excrete in the urine. This avoids the buildup of ammonia. And now we're left with the carbon skeletons of those amino acids for which we can do cool things. If you're left with a skeleton, that is something that is in the carboxylic acid cycle already, or it is pyruvate that can be transformed into something that is in the carboxylic acid cycle already. So pyruvate, remember, we could use pyruvate carboxylase to make oxaloacetate, and that was in the cycle. In these cases, we're able to sustain the cycle, and we're able to sustain the cycle in such a way that we're adding more in than we're taking out. And so we can take out things, we can take out malate, say, to make glucose. We're adding more than 2 carbons. Each turn of the cycle, we remove 2 carbons. So if we're adding more than 2 carbons, that's going to be anaplerotic. It's going to be glucogenic. If, however, we're only being broken down into like acetyl-CoA or acetyl-acetyl-CoA, which can be broken down into acetyl-CoA, yes, the carbons from the acetyl-CoA could enter the TCA, but you're only adding 2 carbons. And if you were to remove 2 carbons each turn of the cycle, which is what you have to do as you go around the cycle, well, then you wouldn't be able to sustain the cycle if you remove something. So we call amino acids that get broken down into acetyl-CoA or acetyl-acetyl-CoA ketogenic, because they can be used to make lipids and ketone bodies, but they can't be used sustainably to make glucose. That doesn't mean that you can't find carbons from them in glucose, because remember, we have a bunch of molecules and they're all hanging out. And if you have the cycle going already, you could kind of feed into it, but you wouldn't be able to sustainably on net make glucose this way. Some amino acids can be used for both because they can be broken down into multiple products, as is the case with some of our branched chain amino acids. Amino acids can also be used to do things like make hormones, make various cofactors, all sorts of things that we can do with amino acids. And many of those things that we can do with amino acids require the